I'm Dr. Fakiraveri, and this is Bacteremia in adults. Here are some reliable resources for this topic. Infectious Diseases Society of America, or RDSA, provides guidelines, and we will focus on IDSA guidelines for bacteremia. First, in 2009, IDSA released guidelines for the diagnosis and management of intravascular catheter-related infections. Then in 2011, IDSA had specific guidelines for MRSA, or methicillin-resistant Staphylococcus aureus infections. This is a wide variety of infections due to MRSA, including bacteremia. So there is a section in there that's specific to bacteremia due to MRSA. There is also a consensus guidance that was published in 2020-21 for optimizing the management of uncomplicated gram-negative bloodstream infections. And more recently, IDSA has a guidance on the treatment of antimicrobial resistant gram-negative infections. The next learning objective is describe the common mechanisms of bacterial resistance to antibiotics. Wild-type bacteria are susceptible to many antibiotics, and they are called wild-type because they have not developed resistance through mutations or acquisition. However, they can still be resistant to some antibiotics intrinsically, and that's what determines the spectrum of activity of antibiotics. Resistant bacteria are the ones that are resistant to antibiotics that normally should have been susceptible to as wild-type. For example, wild-type Staphylococcus aureus is susceptible to penicillin, but you will see that many Staphylococcus aureus strains have developed resistance to penicillin. Wild-type Pseudomonas aeruginosa is intrinsically resistant to penicillin. Now, when bacteria develop resistance, not only can they pass these resistance genes when they divide, but they can also pass it to other species. The transfer of resistant genes occurs generally through 4 different mechanisms. Transformation is when naked DNA transfers from dying bacteria to a competent recipient. Transduction is the packaging of genes, usually from small plasmids, by transducing bacteriophages. One characteristic of bacteria is that in addition to their chromosome, they also have DNA as a circular DNA, which is referred to as plasmid. And this plasmid, because it's small in general compared to chromosome, it can easily be transferred to other bacteria. In conjugation, which is the most efficient way of transferring resistant genes to other species, self-transferable plasmids mediate direct contact by forming a mating bridge between cells. You can see in the picture in the right-hand side the different steps of conjugation. Lastly, there can be conjugative transposition. You see, as opposed to chromosomes and plasmids, transposons are specialized sequences of DNA that possess their own recombination enzymes, allowing transposition or hopping from one location to another, independent of the recombinant enzymes of the host. Here are the common mechanisms of antibiotic resistance. Remember, for antibiotics to work, they need to get to the site of action. This provides several opportunities for the bacteria to prevent antibiotics from getting to the site of action. The first mechanism is reduced permeability. In this picture, we're looking at gram-negative bacteria with 2 membranes, so there's the inner and outer membrane. And for many antibiotics to get to the site of action, they need to penetrate through the outer membrane, often by going through a porin channel. So one thing that bacteria can do is to reduce the production of these porin channels, or in some cases even loss of porin production. And that will basically eliminate the channel of entry for antibiotics. There is also something referred to as biofilm, which we will discuss when we talk about bone and joint infections. Now, if the antibiotics manage to go through the outer membrane, some organisms can actually develop efflux pump and pump these antibiotics out. This is a common mechanism in Pseudomonas aeruginosa. Another mechanism is the change in antibiotic targets by mutation. So if the antibiotics do get to the site of action, then the site of action can mutate so that the antibiotic can no longer bind to the site of action. The fourth mechanism is modification of antibiotics itself. So there are beta-lactamases that can actually break down antibiotics. There are also aminoglycoside-modifying enzymes that can basically alter aminoglycosides. And lastly, there is alteration of metabolic pathways. For example, the evasion of common pathway to make folic acid, which leads to resistance to trimethoprim sulfamethoxazole. Now, there are other mechanisms of resistance, but these are the common ones that we will focus on. Now, let's compare gram-positive and gram-negative bacteria. So on the left, we can see gram-positive, which only has the inner plasma membrane. There is no outer membrane. So on top of the membrane is the peptidoglycan layer. With gram-negative, there are two layers. So there's the inner and outer membrane, and between the two membranes, there is a small layer of peptidoglycan. Beta-lactams prevent the formation of peptidoglycan. So their site of action is where peptidoglycan is. So in order for the bacteria to make beta-lactamase and break down beta-lactams, they need to be made around the peptidoglycan layer. So you can see that for gram-positives, so this will be actually outside of the cell, whereas for gram-negatives, these beta-lactamases will be basically in the periplasmic space between the two membranes. This basically shows you that with gram-positives, they are likely to lose a lot of these beta-lactamases to the environment because there is nothing to hold all these beta-lactamases as they're being made by the bacteria. Whereas for gram-negative organisms, because of the outer membrane, they can hold on to beta-lactamases. So this is more cost efficient for gram-negative. So you will see that beta-lactamase production is a lot more common in gram-negatives compared to gram-positives. ESBLs are an important mechanism of resistance in gram-negative organisms. But before we can talk about ESBLs, let's consider what is a beta-lactamase. So beta-lactam ring is essentially a 4-member ring, and monobactams only have this 4-member ring, and penicillins, cephalosporins, and carbapenems have 2 rings fused together. And what some bacteria can do is that they can make a protein called beta-lactamase, which can essentially break down the beta-lactam ring, and by opening it, they will make the beta-lactam inactive, and that is a mechanism of resistance for them. And there are a lot of beta-lactamases out there. So there are penicillinases that can break down penicillins in general. And then, later on, when we brought cephalosporins to the market, bacteria learned how to make cephalosporinases, which can break down cephalosporins. And later, when we had carbapenems on the market, they learned how to make carbapenemases. And collectively, these are all referred to as beta-lactamases. And of course, to fight that, we also made beta-lactamase inhibitors. And what beta-lactamase inhibitors are essentially compounds that look like a beta-lactam ring, so that they have affinity for these enzymes that bacteria make. And by binding to the beta-lactamase inhibitors essentially make this inactive, so that these beta-lactamases can no longer inactivate the actual beta-lactam, so the beta-lactam can remain active and kill the bacteria. And we have quite a few of them on the market now. We have clavulanic acid, tazobactam, sulbactam, avibactam, weberbactam, and more recently, relibactam. Now, what exactly are ESBLs, or extended spectrum beta-lactamases? So these are the enzymes that can hydrolyze extended spectrum cephalosporins and monobactams. And extended spectrum cephalosporins are essentially 3rd generation cephalosporins, such as ceftriaxone. And they do not hydrolyze cephamycin, so cephamycin are a subset of 2nd generation cephalosporins, and they can also not hydrolyze carbapenems. Now, ESBLs are susceptible to most beta-lactams with the exception of ß-lactam. So ß-lactam does not have good activity or affinity against ESBL. There are different ways to classify beta-lactamases. The easiest one is the Ambler classification that basically classifies them as class A, B, C, and D. Now, from a biochemical perspective, Ambler class B is metallo-beta-lactamase, whereas class A, C, and D have a serine moiety. Class A can be a bit confusing because it has several subclasses. So first we have penicillinases and cephalosporinases, such as TEM1 and SHEV1. And these basically break down penicillins as well as first and second generation cephalosporins. These are sometimes referred to as narrow spectrum beta-lactamases. Then we have extended spectrum beta-lactamases or ESBLs. And these basically are for the most part CTX-M as well as derivatives of TEM and SHEV. And the reason these are called extended spectrum is that in addition to penicillinases and cephalosporinases that break down penicillins and first and second generation cephalosporins, they also break down third generation cephalosporins, as well as aztreonam. Then we have carbapenemases, which basically for the most part is KPC. And what that means is that in addition to penicillinases and cephalosporinases, these also break down carbapenems. Class B, which is metallo-beta-lactamases, are the worst in class, so they basically break down everything except aztreonam. Then we have class C, which is for the most part the AMP-C enzyme, and these break down cephalosporins and penicillins as well as imipenem, but they are stable to many fourth generation cephalosporins as well as meropenem. And lastly, we have class D, which are OXA type enzymes, and these are carbapenemases. So in addition to penicillins and cephalosporins, they also break down carbapenems. Now, over the years, we have developed many beta-lactamase inhibitors that can be bundled with beta-lactamases in order to prevent beta-lactamases from breaking down the beta-lactams. So we have clavulanic acid, which can inhibit class A, including ESBL, but not carbapenemases. It can also inhibit class C, but not class B or D. SS-lactam has activity against class A, but not ESBLs or carbapenemases. It is active against class C, but not class B or D. Tazobactam is active against class A as well as ESBL, but not carbapenemases, and not class B or D. It is active against class C. Avibactam, which is a broad spectrum beta-lactamase inhibitor, is active against all class A, including ESBL and carbapenemases. It is active against class C as well as class D, which is OXA48 type. But it is not active against class B. Weberbactam is active against all of class A and C, but not class B or D. And Relibactam is active against all class A and C, but not class B or D. Now, in addition to their inhibition of beta-lactamases, some of them also have activity on their own against some organisms. Now, the one that's clinically important is that ß-lactam itself is active against Acinetobacter. So this is something that clinically is used. So regardless of what ß-lactam is bundled with, in the United States it's bundled with ampicillin. So we have ampicillin ß-lactam. So the ß-lactam component, but not ampicillin, has direct activity against Acinetobacter and is sometimes used clinically. That was a lot of information, so let's summarize. In general, beta-lactamases can be broken into serine beta-lactamases and metallo-beta-lactamases, and the Ambler classification can be used to classify these beta-lactamases. So class A can be divided into narrow spectrum ESBL and carbapenemases. Narrow spectrum includes TEM1 and SHEV1 among others, and fortunately we have many beta-lactamase inhibitors on the market that can inhibit these beta-lactamases. Now, in addition, there are also ESBLs under class A, and these are CTX-M for the most part, but there are also some derivatives of TEM1 and SHEV1 which have over time mutated, so we call these TEM type and SHEV type. There are many different ones of these, and these are extended spectrum because they can break down third generation cephalosporins as well as monobactams. Now these are pretty much inhibited by most of the beta-lactamase inhibitors on the market except ß-lactamases. Now the worst type of AMR class A are the carbapenemases, including KPCs, one of the most common ones, and there are a select few beta-lactamase inhibitors that can block KPC. Now, just because these are called carbapenemases doesn't mean that they cannot break down penicillins or cephalosporins. They do. Now, AMR class C, for the most part, it's cephalosporinases, AMP-C, and they do break down one carbapenem, imipenem, but they cannot break down meropenem or doripenem. And fortunately, many of these beta-lactamase inhibitors can actually inhibit AMP-C. And the worst type of class serine beta-lactamases are class D. These are carbapenemases as well. And the most common one, or perhaps most clinically relevant one, would be OXA48. And the reason this one is important is that this is the only one that's inhibited by avibactam. Anything else in class D is not inhibited by the beta-lactamase inhibitors on the market. And then overall, the worst type of beta-lactamases are the metallo-beta-lactamases. These are under AMR class B, and these are also carbapenemases. So IMP, NDM, and WIM are common examples. And unfortunately, we don't have any beta-lactamase inhibitors on the market that can inhibit this. Let's take a look at Staphylococcus aureus. So Staphylococcus aureus can be divided based on its pattern of resistance. So the wild type, which is basically penicillin susceptible, is constituting less than 10% of the isolates nowadays. Then we have MSSA, which is methicillin susceptible. MSSA is resistant to penicillin. Then we have methicillin resistant, so MRSA, and the drug of choice for MRSA is vancomycin. Now, while very rare in the United States, there is also vancomycin intermediate Staphylococcus aureus, or VISA, and that is basically that Staph aureus is no longer susceptible to vancomycin, but the MICs happen to be in the intermediate range. And then we have Staphylococcus aureus with MICs in the resistant range for vancomycin, which is referred to as VRSA. Now, MRSA is clinically important, and in general, there are different types of it. So we have healthcare-associated MRSA, and then we have community-associated MRSA. Now, there's also livestock-associated MRSA based on the antibiotics that are used in animals and in food production. But clinically, we basically break these down into healthcare-associated MRSA or community-associated MRSA. And healthcare associated is the more common strain which most healthcare professionals encounter. And this strain is resistant to beta-lactams with the exception of ceftaroline and several other classes, making preferred therapy more broad spectrum agents. Now, community-acquired MRSA is strain only found in the community, and other classes other than beta-lactams are still possibly active against these, so such as like doxycycline, moxifloxacin, trimethoprim sulfamethoxazole. Now, something of note is that the resistance genes that make Staphylococcus aureus resistant to methicillin is the MEK-A gene, which can be identified on rapid diagnostics. And this is the gene responsible for alterations in penicillin binding protein 2A, and that is the target site for methicillin class. So nafcillin and oxacillin, as well as cefazolin, they bind to this site. So when there is mutation, alteration in this, they no longer can bind to penicillin binding 2A, which makes it resistant to that class. Enterococcus species which are gram-positive, can develop resistance to aminoglycoside in 25 to 50% of the isolates. And these are basically plasmid-mediated resistance that are aminoglycoside-modifying enzymes. And of course, there is vancomycin-resistant Enterococcus, or VRE, and there are two main genes that make VRE. So VAN-A and VAN-B, both of which can be identified with rapid diagnostic tests. And basically, the site of action for vancomycin is the alanine D-alanine. And what happens in Enterococcus with these genes is that basically it mutates and changes the second alanine to a lactate. And as a result, vancomycin can no longer bind to the alanine D-lactate. And this mutation basically does not interrupt the life cycle of Enterococcus. So Enterococcus at this point will be VRE. It continues to replicate, and vancomycin will be ineffective. Now, Enterococcus is part of the human flora, so it is common for people to be colonized with VRE, and that's something that can be detected by screening tools on the stool sample. Lastly, let's take a look at E. coli. So E. coli is a gram-negative rod, and it can be resistant to many antibiotics, so including resistant to beta-lactams, and it's primarily through various beta-lactamase production. So including some strains can be ESBL producing, as well as KPC, which is a carbapenemase, as well as OXA48, which is a carbapenemase, and of course the whole subclass of metallo-beta-lactamases that are carbapenemases. So E. coli can be very resistant. At the same time, a wild-type E. coli can be pan-sensitive to many antibiotics. Now, trimethoprim sulfamethoxazole resistance is basically increasing, and it is due to overproduction of dihydrofolate reductase. So dihydrofolate reductase is the site of action for trimethoprim, as well as mutations in the gene DHPS is the site of action for sulfonamide, so sulfamethoxazole. Fluoroquinolone resistance can be due to polymorphisms in the GIR-A, which is a gyrase, the gene for the gyrase. And basically that's the site of action for fluoroquinolones such as ciprofloxacin. Tetracycline resistance can be due to expression of efflux pumps, which is encoded by the TET